So we're studying genetics, and it turns out that genetics is actually really important monetarily to a lot of segments of our economy, such as agriculture, even dog breeding. So for example, if you're looking to get a certain type of dog breed, sometimes it's actually really important that you mate only certain dogs. And so figuring out the genetics of a trait, such as fur color. So here we've got 4 different labs, 4 different color Labrador retrievers. And so it turns out that understanding the genetics is sometimes a lot more complicated than just Mendel's P's. So, of course, here we're talking about genetics beyond Mendel. And so Mendel's principles, we should be able to kind of talk about what he saw in pea plants. But it turns out there's a lot more that affects the appearance of an organism than just dominant and recessive. We talked about nature-nurture, incomplete, codominance, blood type, which has multiple alleles. And now we're on to talk about some other traits, especially ones that don't fit the expected ratio. So I should be able to, even for codominance and incomplete dominance, predict a ratio. It turns out that some are even more difficult to understand. One of them is the sex-linked traits. So we'll get to the Labradors a little bit later, but we actually want to start with the fruit fly, Drosophila melanogaster, D. melanogaster. Is the common fruit fly, and it was studied by a man named Thomas Hunt Morgan. He's who made fruit flies famous, and his whole lab of students. And they were studying the fruit fly for a lot of reasons that we'll study the fruit fly as well. They're small and easy to contain. They have a very short life cycle, so they reproduce very quickly. They also produce a lot of offspring. And some of their traits are really easy to see. One of the traits that's actually easy to see is their eye color. And Thomas Hunt Morgan was working in fruit flies, working, trying to find a mutant. And it turns out that one of the mutations he saw, so one variation, was in eye color. And the wild type found in the wild is red eyes, but the mutant is white eyes. And the mutant is actually going to be recessive. We can kind of predict that. So his theory is that if he mated 2 red eyes, he would occasionally get a white eye. And that if he mated a red and a white, that he should be able to get a half and half. But this is not exactly what he saw. It depended on the gender. So gender plays a role. So in sex-linked trait, gender plays a role because the chromosomes determine gender. Chromosomes that determine gender also carry other genes. So, what this means is that depending on what gender you are more or less likely to pass on certain other alleles. With sex-linked traits, what we see in humans and a lot of other organisms is that the chromosomes that determine gender are not always homologous. So, what we need to think about if we use humans as the example, X makes you female, and that's 2 big X's. Y, big X, little bitty y, makes you male. Having a Y makes you a guy. It's an easy way to remember it. So it turns out that on these chromosomes are the genes that make you female or male. That is very true. But there's also room for other genes. And so for eye color in flies, and also for red-green color blindness, and hemophilia, and muscular dystrophy. We'll talk about these later. You don't have to get them exactly right now. I just wanted you to hear them so when we work problems. But in organisms, there are other genes on here. So they're at a certain location, or what we call a locus, L-O-C-U-S, a locus. The Y chromosome, because it is short, it has actually lost information over time. Because it is short, it does not have that gene. Which means if I'm a male, I only get one version of that gene. So if I'm a female and I have a recessive allele, I could have a dominant allele that hides it. If I'm a male with a recessive allele, that's it. This is what I'm going to show. There is nothing to hide it on the Y. So this is why for me, picturing the X chromosomes and the X and Y is really helpful for understanding what's going on. So in sex-linked traits, which were first discovered in the fruit fly, the chromosomes that determine gender carry other genes, which means that when the parents pass on eggs and sperm, that there's only a letter written on the X. And so you'll notice that we write it X big R for red, X little r for white, and then we have the Y. Those are your possibilities. There's nothing on the Y. And so we write the letters on the X chromosome to show that this allele, this gene, is linked to that X chromosome. And of course what becomes very important is that the male determines the gender. Your dad is what determines your gender. His sperm either had an X in it or a Y in it, and so those got passed on to the offspring. So in sex-linked traits, we just have to keep in mind that there's room on the X. There are other examples in other organisms. So in some other organisms, it's actually the female that has unhomologous pairs. What we're really concerned with is humans and fruit flies. Those are the main examples. Regardless, any time that they tell you that you have an example like this, you would want to be able to locate that the gene is always on the homologous pair. So if we work a problem, a mother is a carrier. Interestingly enough, dads can't be carriers. If they are X little ry, such as this guy, they can't hide anything, they can't carry anything, they are what they are. So we should be able to work this problem. Her husband does not have it. What is the chance they will have a child? The trickiest part about this is actually the wording of the problem. So if we look at the second problem, 6 months later, the mother is pregnant. They find out it is a boy. What is the chance the boy will have hemophilia? So something we have to be careful with is that if they tell us it's a boy, we don't look at the girls anymore. We only look at the boys, and so then we have two choices. So you want to be very careful as you solve these problems. So sex-linked traits will definitely show up. We'll also practice them a lot with pedigrees towards the end of this unit. There are two other things that we just want to make sure that we clarify. First, I want to talk about sex influence. This is where your gender genes impact, so the proteins made by your gender genes, like testosterone and estrogen, impact the expression of others. So testosterone and estrogen can turn on or off other genes, and so when we look at baldness, baldness is not a sex-linked trait. It's not on the X or Y chromosome, but it is influenced by whether your body is making testosterone or estrogen. The other thing is actually a really interesting phenomenon in women, where because we are XX, one of them is randomly inactivated. So that very first zygote that is diploid, one of these will randomly be inactivated and the other is the one that shows. The inactivated is called a bar body. And so this actually is pretty important because it's fairly unusual. The fact that one of the chromosomes is permanently turned off, we call it a bar body. This is actually really important for tracking how genes are passed from mother to daughter and on and on. So, and we're actually able to track the bar bodies. The other part of the notes that we want to cover are some other examples here. For me, the diagrams are really, really helpful. Pleiotropy means that one gene has many effects in the body. So just like estrogen or testosterone. Or another really good example is with sickle cell anemia. We have one gene that does not work, does not function, and there are many effects. So the red blood cell is misshapen, the red blood cells clot, oxygen is not carried. So the idea that one gene doesn't affect just one thing, but that protein has lots of effects throughout the body, and that's pleiotropy. Next we have epistasis, and this is the example with the lab. This is where we have a gene pathway or an enzyme pathway. And so if one gene is not functioning, it affects the whole product. And we talked about this when we talked about regulation. So for example, if A gets turned into B, which gets turned into C, if enzyme number 1 or gene number 1 is not working, it doesn't matter what gene number 2 is. I won't get C. So I could have a functional gene number 2, but if gene number 1 isn't working, I'll never see that C. And the same also applies. So epistasis is where one gene impacts a whole pathway. And so one gene can turn off another. And a really good example is here with the labs. So it turns out that if I have a recessive version of either one, then I don't get the same example as if I had both genes functioning. That the recessive E here, no dark pigment in fur. Even if I have a capital B for dark pigment, it won't matter. I'll never see that. I'll still see no pigment in the fur. Okay. The last one is polygenic. And polygenic is where there are many genes